So we have joining us now for this part of our discussion is Roberto, our first speaker of the day. We also have Shane joining us, Paul, and Majid. Welcome back, everybody, for our panel discussion. Okay. So, reminder to everybody, put your questions in the Q&A section. I do have one or two questions of my own for the panel. I'll open it up to everybody. Today, what's your biggest productivity challenge in your lab? What's preventing you to reach the desired number of samples and throughputs in your lab? Maybe we'll start with you, Roberto.
A lot. We speak about a lot of analysis that at this moment are using the triple quad. In our lab, we perform analysis on environmental samples and for semi-volatiles, all is using triple quad, or non-volatiles like PFAS that Majid spoke before, and also for the food with the veterinary drugs and also the pesticide. All the residual analysis at this moment for the quantitation is performed by triple quad system.
Mm-hmm. And where do you see your largest bottlenecks, Roberto? Is it in the sample prep side, the analysis side, data processing?
No, it's the absolutely is the sample prep. We have used a lot of automations in the labs for the throughput, and there is something that we have found for the liquid extraction, because continue to be a sample prep in devices that we need, and also for questions analysis, some robotics that help to do, and so on.
Okay, very good. Shane, what challenges are you finding with achieving your productivity goals?
Yeah, there's always various challenges, right? As a CRO like ourselves, some days we're doing small molecules, some day we're doing biologics. And so I would say a challenge often is being able to switch instrumentation from running one type of assay to another without having to do a lot of preventative maintenance. Taking an LC that was set up to run a biologic, over the weekend, and then you're flipping it over to run a small molecule the next day. There's a lot of challenges there. I always think that, carryover is a kind of a constant limiting factor. Till we really get rid of carryover, we can't take advantage of the full dynamic range of some of the new mass specs coming out, especially for like PK assessments, things like of that nature. So, that's something we're always striving for, better columns that have less carryover with some of the more complex biologics we work with, and even some of the auto samplers. I think really getting that carryover cleaned up is going to really help take advantage of more of the dynamic range of the mass specs that are coming online. So.
Oh, very good. Paul, I know you work closely with a lot of customers in the lipidomics, metabolomics fields. What do you see as their major challenges for productivity?
Yeah, I think that it's an omics type of experiment that we tend to do, and there has been a general trend in lipidomics and metabolomics to move towards targeted assays, just because of the challenges that we face in with software interpretation of discovery data. But where I think where I'm excited about is that the challenge is that you don't know what you don't know. So if you have a targeted list and you're not looking for something, then you're not going to see it. So what I am kind of excited about is, say, for example, the 7600 system that has the ability to do targeted quant, but at the same time, you can take that data and go the extra mile and start doing some discovery work. Electron activated dissociation is pretty much the biggest innovation, in my opinion, for lipid structural characterization that's come out commercially. So, I mean, I think that it's a balancing act. How many molecules are we going to look for? And if we don't know what that we should be looking for, how do we approach that? But it's neat to think that there's an instrument now, or their instrumentation, the technology has gone to the point where you can do both types of experiments on the same instrument and not have to really have two different workflows and different streams of work.
Okay, and Majid, same question to you.
Sure. I think, the biggest challenge, when it comes to PFAS, it's known down there is that controlling the contamination within lab space, like having the control environment, it's the things that everyone is talking about. It's easy to bring the method up and running, but keeping the method and meeting all the QC requirements and having a control method, it's a big challenge in the lab. Knowing the PFAS could come from the sample prep or the LC analysis, it's making it even more challenge, especially troubleshooting the method when you have contamination. So I think, within PFAS in particular, contamination, cross-contamination, and having a control environment is the biggest challenge in the lab.
Oh, very good. Yeah, so we got a different answer from all of our speakers, depending on what their actual workflow is. I think that's the beauty of the panel we have today, is this diversity of techniques. There maybe, Majid, a couple of questions for you that we didn't get to. The bottles that you use for the water collection, were they a special type of bottles, or what guidance can you give to people to use for that sample collection?
So we purchase pre-reserved water bottles from the vendors. So there are vendors down there who are selling these bottles with the Trisma or the ammonium acetate in it. So we purchase the bottles from them, but we also do like in-house testing. So every time we're receiving, like, let's say we order like 10 cases of these bottles, and they come with different lot numbers. So we pull sample from each of lot numbers, and then we test them for PFAS to make sure they are PFAS free before we use them for any collection. So, vendor-wise, there are a couple of companies down there that are either selling empty bottles or bottles with a preservative in it. So the one we're using is just like bottles with preservative in them. So we purchase them, we test them in-house, and then we use them for sample collection.
Oh, very, very good. Question for you, Paul. Your method showed off polarity switching features of the 7500 plus. Someone asked, typically in a single polarity experiment, you're picking the mobile phase additive, right? That's going to give you the best ionization for that polarity. When you're doing concurrent positive and negative switching, what sort of compromises or changes do you have to make there in your selection of mobile phase additives?
Actually, it's fairly straightforward with lipidomics. We use ammonium acetate. So when we're detecting neutral lipids, such as triglycerides and diglycerides, we look for the ammonium adducts. In the negative ion mode, there's really only one particular phospholipid we need to be concerned about, which would be phosphatidylcholine, and we look for the acetate adduct. So the nice thing is we can do both with that with the ammonium acetate.
Oh, very good. And Roberto and Shane, I know you do analyses in both polarities. Any comment or answer to that question about mobile phase choices?
No, I would say for oligonucleotides, you're most likely doing negative ionization. And so we're always choosing, we're somewhat limited. We always have to have the ion pairing reagent in there to get that oligo to retain to the column. So the biggest thing for us is just, finding that perfect balance of enough modifier, you get good retention, but not so much modifier that you're losing sensitivity at the mass spec. So that's more the issue for us. But we tend to only run negative ionization just because of the nature of oligos.
Okay. And Roberto, what's the mobile phase modifier that works best in your methods?
And no, but we use also acetic acid or formic acid, one of the two.
Okay. Majid, a specific question came in for you for PFOS analysis. Which solvent is best for the standard stock preparation?
So with so since we're following EPA methods, I mean, we just follow exactly what's written in these methods. So when we do the sample, the color prep for the standard reference materials, we're using 96.4 for 537.1, and then we're using 80 to 20 methanol on water for the 533 to prep the standards.
Okay, very good. I did mention, the diversity of techniques that everybody is using, but everyone on the panel has, one thing in common, right? That's quantitation by LCMS. And I know during the last few days, we have had many young scientists and trainees joining us. I wonder if each of you could just talk a little bit about, your journey to your current position, right? The sort of training that you got that made you successful where you are now. Is this a planned route? Did you start out in a different scientific discipline and then migrated to LCMS? Majid, maybe we'll start with you. Hear a little bit about your scientific journey to where the position you are now.
Sure. I got my PhD in organic chemistry, but I did a lot of analytical instrumentation as well. So I never thought one day I will be working in a public health lab, doing a lot of environmental testing. My vision more into joining big industries and maybe pharmaceutical companies and work as a chemist, not environmental. But I graduated in 2019, and that's 2020 when I started looking for a job, when COVID started to be a thing. And then all the attention went to the public health lab. So that brought it to my attention. I mean, maybe possibility to join a public health lab and work under environmental testing. So I joined down here and I liked it. I mean, the things I like about doing the things in here, it feels that whatever that you learn chemistry as a chemist, you bring that one in use to help your community addressing a lot of emerging contaminant. It feels that your experience, your knowledge, it's something useful for the community. So that when I start doing a lot of LC mass spec and gain more skills and knowledge within that field. And here it goes. So it's 5 years dealing with the PFAS and things keep going.
Very good, Majid. Maybe I'll just stick with you for one next question. Where else do you foresee that we as a society and you as a public lab going to have to be testing for PFAS? Given its ubiquity. Besides the water that you went into detail on, where else are we going to have to be testing for PFAS?
So testing for PFAS in water has been around for a couple of years. Now the things start to be more into PFAS in biological sample. I mean, we all know that PFAS in the water, and now we all know that we got exposed to PFAS at some level. The next question is, do we have them in our body and to which level that we have them? So I would see that the focus coming up is toward more of a biomonitoring, like where we see the PFAS in the food and the biological sample, blood, serum, and plasma. So that's going to be the next thing.
Okay. Shane, maybe I'll go to you next. Maybe you can talk a little bit about your background and your journey to where you are now using quantitative LCMS.
Sure, yeah. So, my first job out of the university, I was doing synthetic chemistry. So I was working at a medical device company, and I was doing, in their research and development group, working on different homologs of cyanoacrylates that we were working on. And so, through that, I got to learn the importance of analytics and being able to take the experiments you ran and, run those and actually, get some information to take that information back to the lab and do your next set of experiments based on what you saw. And over time, I just kind of migrated more towards the, analytical side of things, a little less away from the synthetic side. And then, somewhere through the career, I got invited to come to, what was Pixon Labs, now Larry, and just asked to help set up a bioanalytical lab. And so, I didn't have a lot of experience with it at the time. This was in like 2001, but I just took the opportunity and figured, I'm sure I could figure it out, fake it till you make it kind of thing.
Yeah.
And, over time we built up a mass spec laboratory, and now we're, 200 plus people with 4 different sites. So, I guess what I would say, learning from there is just, when people ask you something, just say yes and give it a try. You never know what you can accomplish. Don't just say, oh, I've never done that. I don't think I should, put myself out there. Putting yourself out there is where you discover a lot about yourself and, people around you. So.
Yeah, that's very similar to my first experience with LCMS. Started at a very small CRO as employee number 2.
Yeah.
And, my boss just said yes to everything. Can we do peptides? Yes. Can we do oligos? Yes. And it was up to me as the lead analytical scientist to figure it out. Those are the most challenging, but most exciting, job experiences you can have. Shane, specific question to you. Someone asked the ion pairing agents that you used. Are they harmful? Do they contaminate the mass spec? How are you managing that?
So we find that they don't contaminate the mass spec near as much as they contaminate the LC system. And so if you're taking, so we tend to have designated LC systems that we run ion pairing reagent on. Otherwise, we have a cleaning protocol where we have to, run things like water and acetic acid solutions to really get rid of those ion pairing reagents after they've been used, before we could switch over and run like another type of assay, like a small molecule or a peptide. As far as the mass specs, they tend to not have as much problem with the ion pairing reagents. We always wipe the curtain plate, in between runs, but typically all of our problems are really on the LC side when it comes to the ion pairing reagents, and they do stick around if you just, you can't just swap the systems without doing some type of pre-cleaning. And, so it's best just designate a system for ion pairing reagent and save yourself a lot of headaches.
Oh, thanks for that, Shane. Roberto, we'd like to hear a little bit just about your journey to your position.
It's long. I started in '89 and I worked with a company as a customer engineer for a long time until the 2016, and for repair, maintenance, installation of instruments from GC line, LC line, and so on. Then I moved to Labs as operational manager, and I working to build this lab for the futures, and now we started that we was 40 people, and now we are 150 in 10 years. We have a nice goal. We had the food matrix in our portfolio, the food contact material. We have grow fine. It was nice because I built something and I don't installing only an instruments.
Yeah,
I see whatsoever is around.
I know Biochemie Lab also has expertise in PFAS analysis. Maybe I'll ask you, Roberto, where do you see that you're going to have to go next for testing for PFAS?
For PFAS, we move. We start with a method that was we use the STM for the waters, and we perform analysis by direct injection, and with a nice sensitivity for respect our European law. On the other side, the law changed. Now we need to go to 0 PFAS, and that means that we need to have less LOQ. And now we are working with SPE to use exactly the same instrument method, but have a good concentration of the samples both online and offline. And probably we'll approach the offline for cleaning matrix like drinking water, and we use the offline for dirty matter, wastewater, and so on.
Oh, okay.
On the other hand, we start only to working with PFAS with food, inside in the food.
Okay. Paul, let's move to you. Be interested to hear your journey from where you started to where you are now.
Yeah. Well, it's I have to say that I've had kind of a strange journey, but it's always been unified by really my passion for lipids. My father was a lipid biochemist, and I started working in his lab when I was 13 years old, scraping TLC plates and playing with radio label. It probably explains my curly hair, but I've kind of followed that pathway. I went to undergraduate. I did a degree in chemistry and English, and I went to grad school in biochemistry, working with lipid biochemists there. I really followed the academic pathway. I was an assistant professor at the University of Pittsburgh School of Medicine for about 7 years. I was unfortunate enough to be kind of earning my chops during probably one of the lowest periods of NIH funding. And I honestly just got tired of the game. So I had an opportunity to come to SCIEX, and it took a lot of hard decisions, but I did it, and I haven't looked back. I did take a little what I call my sabbatical, and I was a director at Avanti Polar Lipids for several years. But I'm back at SCIEX, and really my whole career here at SCIEX and in academia has been figuring out new ways to look at lipids, discovering new lipids, and it's been very satisfying. And now I'm in charge of all the scientific content that comes out of SCIEX with regards to lipidomics and metabolomics. So it's I landed nicely, but it's been a fun journey, I will say that.
Yeah. Okay, we did get a couple of specific questions come in for you, Paul, about your methodology. It asked earlier about the additives you're using in your mobile phase.
Mm-hmm.
We're getting more questions for you to be a little bit more specific, in terms of the concentration that worked best for that polarity switching method.
Yeah, I typically use 10 millimolar, which is one of the offered suggestions there. I did want to add just a little thing. Helichromatography is very susceptible to changes in pH, right? So it's one of the reasons why you have to do an unscheduled test for every new solvent batch you make. We found recently that if we don't adjust the pH, we just keep it at the pH that it comes out when you mix up the solvents, our retention times are much more stable, right? But that being said, that can affect your ability to see some molecules. Like, for example, if we have a high pH, which is what's typically recommended for helichromatography, we get really nice, robust cardiolipin signal and BMP signals. When we don't pH it, then it's harder to get those because they're multiply charged in the negative ion mode. And so we find that having slightly basic. So it's a balancing act. When you're trying to measure 2,500 compounds in a single run, you're going to have some compromises. And so if you do need to adjust the pH to make it basic, ammonium hydroxide is preferred. But one thing I want to point out, and this is something that I think is a challenge for those people who have migrated from, say, proteomics or metabolomics and go to lipidomics, is they tend to like to use formic acid. It obviously does great enhancement of the chromatography, but what I would suggest is to use acetic acid. It has a pKa of around 4.5, which is the same as the fatty acid you're looking for, whereas formic acid has a pKa around 2.5. And so it's competing for your signal. And if you're in the negative ion mode and you need to have an acidic pH for doing lipid mediator analysis, use acetic acid. You'll find your sensitivity increases about 5 to 10 fold. So, those are the tricks that you learn. And a good question. I appreciate it.
Okay. Maybe I'll just, we'll take a minute for one last question to our industry panelists. Where would you like to see a scientific company like SCIEX, specifically innovating or developing to meet your needs for the future? Roberto, why don't we start with you?
You start just with the plastic instrument now, because when I I think that in Europe we bought the first instruments, the first 7500, and we see the sensitivity. Now we are seeing also the robustness of the instrument. We have done a lot for the moment.
Okay. Very good, Roberto. Shane, how about you?
Yeah, for me, I think the question we're always trying to answer is what's in the sample, how much of it's there. And so, that's where you really need, I think quantitation is great with triple quads, but you really need some high res instruments to answer some of those, what's there questions. And so I guess what I think everybody's always going to say is better detection limits, right? Everybody wants to see further down. So that's always important. But the other thing is we continue just to see new molecules, right? So now we're looking at, it used to be probably 80% of our work was small molecule. Now it's probably more like 70% is protein-based work. Small molecules are still around, but proteins have a lot more challenges. And I think with those, especially detection limits become more and more important, especially if you're looking at, like mRNA drugs and you're trying to figure out, do they actually translate proteins? How, did you see a human protein translated into a mouse when you're, evaluating new mRNA drugs? So trying to go to a lower detection limit is important. And then getting that specificity, selectivity, so you can tell like a human protein that is being transcribed in a mouse is very important to us. So I think what you guys have done with the 7600 Xenotoph is amazing. I'm excited to see the 8600 or 9600s as those come and what you guys achieve with that. So I just keep pushing detection.
Yeah, well, we certainly didn't intend to launch any products or create any future products today, but appreciate your optimism for the future there, Shane. Majid, let's go to you for our final answer for today.
Sure, I will go with Shane. I mean, the more instrument that will give us the ability to see like even lower than what we're expecting to see, it's the things that we're looking into these SCIEX instruments. We have used a triple quad 6500. I mean, just to give you an example, we can see a very good quant limit when it comes to PFAS. We're looking into getting the 7500 plus, hopefully, so that would help us even lower the quant limit for some biological matrices. But I mean, again, the need is to keep building these features into the instrument that will enable lab and scientists to even like target a very low quant limit for a lot of like emerging contaminants with different matrices.
Oh, very good. Thanks, thanks, Majid. Okay, we're out of time for today. So I'm going to thank all our speakers today for joining us. I know your time is very valuable, and we really appreciate you giving up your time for us and for everybody online today as well. So thank you very much, and wish you all the best. Goodbye now.
Thank you.
Thank you.
Thank you.